Hello everyone and welcome back to our session dedicated to fundamentals of nucleic acids. After we had a look at the structure of DNA and RNA, let us have a look how those molecules within DNA are actually arranged. So the bases in DNA are joined by hydrogen bonds. Those bonds are individually actually pretty weak. However, nucleic acids can be quite long, so collectively those bonds are very, very strong and provide the molecule with stability. So you can see here the individual strand of a double-stranded DNA molecule is arranged as we discussed before, with the 5' and 3' end on one side, but we also do have an antiparallel strand with 3' and 5' end here. And this is actually one of the very, very important properties of the DNA, more specifically of a double-stranded DNA molecule. The individual strands within this molecule are antiparallel. What's also quite obvious here, that the bases are paired always with a specific partner. We have all of the A's paired with T's. However, in RNA, this pairing looks different. So there are indeed double-stranded RNAs, especially in viruses, but they do not occur in human cells, where A is actually paired with U. However, for you, it's only important to know that in human DNA, A is always paired with T in DNA. In contrast, G is always paired with C. And as you can see here, there is actually a pattern. So the purine is always paired with a pyrimidine. And this is quite nice thing to remember. Additional property of the double-stranded DNA molecule is that those strands are actually complementary but not identical. And this makes sense. So the sequence is different and those strands are antiparallel. So please keep in mind they are complementary but they are not identical. The question for you guys here would be, based on the knowledge from this session, imagine you are working in a DNA analysis lab and you picked up a species from a patient sample. So you analyzed the DNA and found in the sample that 20% of the bases are adenines. So what percentage of those bases would be cytosine based on the base pairing rules? The obvious answer here is 30%. But why? Obviously, if we add all of the bases in a molecule of DNA, then we need to end up with 100%. Because we know that A always pairs with C, the percentage of T must be 20% as well. To get 100%, we now need to have the whole molecule as 100% and subtract 40 from 100. Then we end up with 60. If we divide it by 2, then we end up with 30%, because the amount of C and G is always the same. We know actually quite a lot about the double helix and how DNA looks like in detail. And in order to share with you guys how exciting the discovery of the double helix actually was, I would like to show you a very short movie to make you familiar with the history of DNA discovery.
As late as 1951, the chemical structure of DNA remained a tantalizing enigma. At Cambridge University in England, biologist James Watson and physicist Francis Crick had been working to unlock the secrets of the DNA molecule. But they weren't alone. Several other groups of competing scientists were hard at work trying to solve the same puzzle. Some important facts about DNA were already known. For instance, scientists knew that DNA was composed of 4 bases: adenine, cytosine, guanine, and thymine. They also had been able to infer something about its structure with the help of X-ray crystallography. This technique involved passing an X-ray beam through a crystallized DNA molecule and capturing a vague, shadowy image of its internal structure on a photographic plate. Armed with this information, along with their own knowledge of chemical structure, Watson and Crick began building a 3-dimensional model of DNA.
I wanted an arrangement, you know, where I had a big and a small molecule, and somehow you had to form link bonds. Here's A, and here's T, and I wanted this hydrogen to point directly at this nitrogen. So I had something like this. Oh. So then I went to the pair, and I wanted this nitrogen to point to this one. I went like this. Whoa.
Today, you can buy a kit and assemble the structure that Watson and Crick put together. But we're getting ahead of ourselves. Watson and Crick needed more data. With the help of physicist Maurice Wilkins, they gained access to an X-ray picture of a DNA molecule that had been taken by his research partner. Her name was Rosalind Franklin. Without her knowledge, Watson and Crick used the data from that X-ray image and successfully completed their model. In a 1953 issue of Nature magazine, Watson and Crick revealed their amazing discovery to the world. Their model showed that the DNA molecule was a double helix. The twin strands were composed of pairs of the 4 known bases linked together by hydrogen bonds. And the whole structure corkscrewed like a spiral ladder, which could easily pull apart in order to make copies of itself with the same encoded genetic information. Watson and Crick had won the race. The discovery of the structure of DNA sparked a scientific revolution. It illuminated the molecular and biochemical foundation of life in a whole new way. It opened doors for areas of research and other great discoveries that few ever imagined possible. As for Watson and Crick, their discovery of the double helix, along with Maurice Wilkins, won them a share of the 1962 Nobel Prize. And what of Wilkins' colleague, Rosalind Franklin? Despite her contribution to the discovery, she wasn't considered for the '62 prize. The rules state that it can only be awarded to a living recipient. Rosalind Franklin died in 1958 of ovarian cancer, most likely caused by exposure to X-rays.
As you can see, science can be not only interesting, but from time to time also a bit cruel. So you might actually ask the question, what Watson and Crick discovered in reality? And some might say Rosalind Franklin's notes, and I would agree. But back to serious business. So next question for you guys is, which technique was used to determine the double helical structure of the DNA? And the correct answer is obviously X-ray crystallography. Good. After we have learned a bit about the structure, let us have a look at the DNA helix as a drug target. So this is something quite important for you as future pharmacists. If we look at the Watson and Crick structure in detail, then we will see that there are actually two types of grooves in this molecule, the so-called major groove, because it's quite large, and we also do have a minor groove, and this is simply due to the intrinsic structure of a double helix. You always end up with one major groove and minor groove. So this is because the backbones of the strands are closer together on one side of the helix than on the other. And the major groove occurs where the backbones are far apart, and the minor groove occurs when they are close together. In drug development, indeed, many drugs are designed to interfere with both of those structures, especially anti-cancer drugs, because cancer cells tend to divide pretty rapidly, and in uncontrolled manner, drugs they are binding to DNA are actually quite useful to prevent replication of the DNA, thereby slowing down the progression of cancer. Good, so we have already learned that DNA is the basic molecule of heredity. There is only one type of the DNA in our cells, and it is also the carrier of hereditary information. And the evidence for this is because DNA is present in all cells and virtually restricted to the nucleus. With one exception, there is a bit of DNA present in mitochondria as well. Also, the amount of DNA in somatic body cells of any given species is constant. That means if you would isolate DNA from nerve cells, the amount of the DNA would be the same as the amount isolated from, let's say, muscle or liver cell. This is quite solid evidence. In contrast, the amount of proteins varies between different cell types. So the question here would be, how could we prove experimentally that DNA is indeed molecule of heredity? And some of you might actually have heard about those experiments in the school already. The first proof of DNA being the molecule of heredity has been provided by Frederick Griffith as early as 1928, and in his experiments he used pneumococci. And this is basically a germ that causes deadly pneumonia. Pneumococci do occur in two flavors. We have the rough colonies and also the smooth colonies. The R strain is benign, so this strain lacks a protective capsule, can be recognized by the body's immune system, and it gets destroyed fairly easily. In contrast, the S strain, which you can see on the right hand side, is virulent. That means it can result in disease because it cannot be recognized by the immune system. In the Griffin experiment, those guys isolated S strain pneumococci. So again, those are the bacteria that cause disease, and infected mice with this type of pneumococci. As a result, obviously, the mice got infected and died. What they have done then, they isolated the pneumococci colonies from the infected and dead mouse, and were able to isolate and expand new bacteria. They also injected R strain pneumococci. Those are the pneumococci that do not cause the disease into the mouse, and the mouse remained healthy. They have been then able to isolate R colonies and expand them. However, if Griffith he'd killed S cells, those virulent cells, and infected mouse with them, the mouse remained healthy and no colonies could be isolated from those bacteria. In most crucial and important experiment, they, in addition, mixed living R cells, those are the bacteria that are not able to produce disease, and combined this with heat-killed S cells. Here, the mouse actually did contract pneumonia and died. In the following, Griffith has been able to isolate both R and S colonies. This means that there must be a transfer of genetic information between living R cells and the remains of heat-killed S cells, and also they could nearly exclude proteins because proteins are heat sensitive. So something from the dead S cells has transformed the R cells to become virulent. They also found that progeny of the transformed cells were S-like. This was already a very good evidence that proteins are not the carrier of the genetic information, but the final proof that DNA actually does the job has been provided in 1944 by McCarthy and MacLeod. They used the very same model system, they used S cells, and they heat killed them. In the following, they produced a suspension containing lipids, carbohydrates, and basically all of the components that have been present in the cells before as well. The sample has been then subdivided into different wells, and in one well, they added proteinases. Proteinases are enzymes that destroy proteins. So this sample did not contain protein. In another well, on the container, they added ribonuclease, which gets rid of RNA. So this sample did not contain RNA. And in the last sample, they added deoxyribonuclease, an enzyme which destroys DNA. Then they added R cells to the mixture, and finally realized that even if no protein was present, R cells have been transformed into S cells. So this excluded proteins once again as a carrier of genetic information. Interestingly, in the sample that did not contain RNA, if R cells have been added, also the S cells appeared. That means that RNA is not the carrier of genetic information. In contrast, if no DNA was present in the well, no S cells appeared if R cells have been added. So, to conclude those experiments, the transforming factor must be DNA, and therefore DNA must be the carrier of the genetic information. With this, I'm ending the second session of the lecture, and I will see you soon in session number 3. Thank you very much for your attention.